Okay, cool. So hi everyone, I'm Jiazhen. I also go by Jojo. So both Bo and I will be your TA for your homework 3 and homework 4. So before our lab starts, I want to make several announcements. The first thing is remember to fill out homework 2 feedback survey. Homework 3 is due on Monday instead of Sunday night because of the wellness day. And also we have uploaded a new version of homework 3 this morning, actually this afternoon. And please do not work on part 4, sorry, please do not work on part 5 and part 6 yet until we make an announcement that all the data sets are finalized, okay? And if you have any question, always feel free to post in the Slack channel. And something interesting about the homework is we actually have 2 bonus questions at the end of homework 3. And it's more like redesigning the homework questions for next year students. So if your solution is selected for the future homework, you will be granted 5 points. And besides, we have office hour every Friday and every Saturday. I would definitely encourage you to come if you have any question. And definitely start early for this homework. It's not like it's very complex, but there will be a lot of bash commands, and some commands would require you to submit on a web page, and those web pages sometimes can be slow if a lot of people are using it. So I would suggest you start early for this homework, okay? And let's do a quick review on ChIP-seq. I think Shirley also talked about ChIP-seq in this morning's pre-recorded video, but the key idea is we want to cross-link protein and DNA into the status of binding together, and then we can pull down the protein that we're specifically interested in by adding an antibody that only binds to the protein we're interested in. And then you can sonificate the DNA and protein into smaller fragments and then do PCR to enrich the DNA fragments and then send it for sequencing. So there are non-histone chip and histone chip. Non-histone chip is more like targeting transcriptional factors binding, while histone chip is more like trying to find histone modifications. And here is an example of how ChIP-seq signal would look like. In the left figure, the ChIP is actually immunoprecipitation followed by microarray, while the middle figure is immunoprecipitation followed by sequencing, which is our data set for this homework. There's also, you might also heard about control. It's just not adding antibody, but still do the same procedure of sonification and PCR and send for sequencing as a control or as background signal. So on the right, it's an example of different protein chip signal. So for example, CTCF is a transcriptional factor. So you would see very sharp signals at very specific genome location. That's because transcriptional factor like CTCF, they only bind to unique DNA patterns. And for RNA polymerase II, you will see a much wider peak signals. And for the later two, it's histone modifications. It's much wider and much deeper signals compared to the transcriptional factors. And we keep saying peak calling for ChIP-seq because, as you can see, it's like small peaks, right? When there is a binding event at a specific location. Cool. So now I'll go over the part 1 of homework 3, which is about the initial processing of ChIP-seq data. And we use a lot of MACS. And here is a general workflow of ChIP-seq analysis. We send ChIP-seq library for sequencing, and company like Illumina would return us FASTQ file. It's also the starting file for your question 1. And then we want to align it, align the FASTQ file to human genome, and which will give us BAM file. And then we may want to do some quality control here and then perform peak calling using tool like MACS2. and that will give us a bad file. You might want to do quality control again. And then for downstream analysis, there will be motif discovery, co-interacting TF finding, and also maybe integrate with RNA-seq data and so on. So there will be a lot of downstream analysis of ChIP-seq. I'll also go over bash script just because we will use a lot of bash script in this homework. Here is your bash script header. You can actually adjust the running time and running memory. So time is here, it's set in minutes. If you want it to run longer, you just set it to a longer time. So the same thing for memory. But just to let you know, if you make it too long and too large memory, the server might actually put your job to later queue. So you might wait longer for your job to run. And here, /o is your output file and /e is your error file. So no matter what should be output to your terminal will actually be output to these two files. You can also change the name, so you can take a look at those files later. And then we also received some worries about which module to load. I mean, we will cover it in this lab as well, but when you are unsure in a new environment, you can always type module avail, or you can type module spider followed by module name, which will give you which packages you should load together with the package. And then to submit your job on a Slurm server, you just type cbatch, your script name, and then to check the progress, you can use sacct. And if you want to cancel the job, you can just do cancel job ID. If you want to know more options about Slurm, because Harvard server is built based on Slurm, I think Slurm is also widely used in many other systems. So it would be nice to maybe learn more about it in your free time. And then we also require a lot of screenshots in this homework. Here is just how you can insert figures in your RMD file. Yeah, I think I can go next slide. Oh, sorry. So now let's talk more about MACS. So MACS is actually a tool developed by Dr. Shirley Liu's lab in 2008. And I think it's definitely one of the most standard and most widely used tool for analyzing ChIP-seq data. And actually, even in recent years, when people are doing single-cell ATAC-seq analysis, I would see their pipeline calling MACS to do peak calling for ATAC-seq, single-cell ATAC-seq as well. So I think it's definitely a very nice tool to learn about. And I think Shirley already covered how MACS work in math, like its mathematical model and why they develop it in this morning's lecture as well. I mean, in the pre-recorded videos. So for me, I will just talk more about how to actually use it. And there's a list of functions available on their GitHub page. I pasted on the left side. But the key functions relevant with our homework 3 is first callpick. It's the main function for calling picks. And then is filter dupe, which can be used for removing duplicate reads. Maybe we can take a look at their page. Can you guys actually see the GitHub that I'm opening?
Yeah.
Okay, thank you. Okay, cool. So let's click on callpick, for example. I just like how they give you a description of like each option, and it's also nice about they actually give you, let me go there. They actually give you what will be the output files, because for your question 1 to 4, I think sometimes students get confused about the output because there will be 4 to 6 output files. Just to let you know, the one we'll be mainly using is the name submits.bat file. But there are a lot of other statistics, such as log fold change in the narrow pick as well. So if you're confused about what's actually in each file, you can take a look at this GitHub page, okay? So now let's go back. Okay, so for question 1, for question 1, it's asking you to align the FASTQ file to human genome. And we want you to use BWA, which is a very fast aligner. So if you check BWA page, there are different options. We recommend BWA MEM because it's for reads longer than 70 BP. There are also options like BWA ALN, which is for reads smaller than 70 BP, I remember. So I think in our case, BWA MEM would work better. And for your bash script, don't forget to load required module. And then we ask like, what proportions are uniquely mapped. So you can use SAMtools for getting those summary statistics, and then just take a look at your output file that I just mentioned. I think those statistics will be output in those .out file. Okay, so that's for question 1. So for question 2, so now we already align your FASTQ to human genome, and we got some BAM file. We want to use MACS2 to remove the PCR duplicates because in PCR amplification, some reads might get amplified much more compared to others. And so we want to keep, so here's like keep dup 1, which means we'll only want to keep 1 read of those duplicates. And again, don't remember to load CentOS 6. I think it's required for MACS2 to work. And then the filter dup /I is just passed to your input BAM file. And then keep duplicates is how many reads you want to keep. /O is the name of the output BAT file you want. Okay. And then we also ask you what's the percentage of reads that are redundant. You can actually find that in your .err file. So there will be a lot of lines printed, and there will be one line saying, maybe I can show you guys in the terminal. 1 second. So can you guys see my screen of the terminal commands? So, yeah. Okay, thank you. So there will be a line saying redundant read of alignment file 0.17. So that's like the percentage of redundant reads in your BAM file. Okay. Now let's go back. Okay, cool. So that's like a part of the quality control process. And even though I know this question is only for graduate students, but I think for undergrad, in order to do Q3, Q4, you also need to do Q2 as well. Okay. And then for question 3, it's also like quality control, as Shirley talked in lecture, where we want to further filter the peak results by FDR and by fold change. So I think she said like, in the past when there's really small amount of reads available, the fold change is set to 2. But, as nowadays sequencing technology gets more and more powerful, you can set fold change to much higher number. So in this question, we're asking you to set it at 5. And again, load the required packages. And so now we're using the call pick function to perform the pick calling step. And you can set the FDR by the /Q, and you can change the fold change by setting this FE cutoff option. So on the bottom, I list like what does each command mean and what you should put there. So I think if you follow this, that should be fine. And then we ask how many picks do you get from each condition. So you can just do a additional WC/L, which is word count by line of your of your output file. And as I said, it will output multiple files. And the one we want to use for downstream analysis is submits.bed. And it should open with this prefix. For example, if you put /n Q3, then your output would be Q3 / submits.bed. So that would be the bed file we want for downstream analysis. And you want to do this for both the normal sample and the tumor sample. And also, by the way, so for our homework, we're using AR ChIP-seq. We're specifically pulling down, I think, androgen receptor in prostate sample, both prostate cancer and prostate normal sample. Because I think in lecture, Shelly also mentioned how estrogen get, upregulated in breast cancer sample. So it's a similar idea. We're just looking at androgen receptor ChIP-seq results in prostate samples. Okay. And for question 4, so by the way, in this step, MACS2 can perform peak calling without a control. So that's why we only have /t here, but we don't have /c because we don't have a control file. The normal sample is like normal sample. It's not the background DNA input control. But in question 4, we are saying, since we actually don't have that control file, you can just use normal sample as a control. And that's why we're doing /c. You can just put your normal file here, okay? And then you can just change FDR and fold change to corresponding value in this command. So I think that's about part 1. Do we have any question for now? Okay, so now let's talk about the part 2 of your homework 3. It's still about quality control and about how to use Systrom data browser. So for question 6, it's asking us to use Systrom data browser to check public available ChIP-seq data. I'll just do a live demo of this. Okay, so can you guys see Systrom data browser now?
Yeah.
Yes.
So we're looking at Homo sapiens, and we're specifically looking at AR ChIP-seq. That's our transcriptional factor, and we want to check prostate. Yeah, it takes me a while to find this. We're specifically looking at prostate. Actually, you might need to click multiple times to get all 3 selected. Okay, cool. Now we get all 3 selected. And you can see these are actually some public data available from previous literature. Here is like the quality of their data set. And I think question 6 specifically asks us, like, do we have over 100 AR ChIP-seq that pass all quality control? So you can set samples passing all quality controls here. So that means they pass all 6 quality control, okay? And I think since one page has 20 samples and there are 9 in total, we probably have over 100. We're fine with a screenshot, but if you want to get exact count, we're fine as well. Okay, so that's for question 6. Let's go back and see question 7. So does transcriptional factor, oh yeah, can you comment on what QC measures tells you whether a ChIP-seq is of good or bad quality, and have a screenshot of each example? So I guess let's go back to the Systrom data browser again. Yeah, actually, after you choose that option, you probably need to refresh the page and then reselect Homo sapiens, prostate, and AR. Okay, cool. So as you can see, for those, if you click on it, you will actually give you more details about the data set at the bottom of the page. And if you hover your mouse over, it will show you what's each quality. So green means passed the quality control and red means didn't. And as you can see, even for some data published on Nature Communication, it still might not be good quality because here you only pass 2 quality control. So I think you can just maybe pick some and take a screenshot of 1 good data set and 1 bad data set. And then maybe out of the 6 quality control, pick some criteria that you think are the most important ones for ChIP-seq QC check. I mean, it's more like free response. You can put whatever answer you like there, but maybe just choose the one that you think are the most important QCs and have some explanations, okay? So I guess now let's go question 8. Oh yeah, so it's still about using Systrom data browser, and it's asking like, what companies have made this AR antibody? Because to do ChIP-seq experiment, we need the antibody that only binds to AR. And if the antibody is not working very well, like if it also binds to other protein, then the ChIP-seq result won't be very good. So you can, so for every experiment, for example, you can maybe take a look at some good ones and click on it, go to the bottom. Here there's GEO or ENCODE, click on it. Okay, cool. So here on the characteristics, it will actually show you the antibody name and which company made it. Oh, okay, cool. So this is for some good quality ones. You can also maybe just look around and check some bad qualities and see whether maybe the ChIP-seq quality is relevant with the antibody company, or maybe with some specific product, or maybe not. Again, this is like a free response question. You can just give your thoughts here, and we're open to any answer. Okay, cool. So that's for part 2. Do we have any question here? Okay, so as I talk about in part 1, we're like doing the alignment and quality control of our AR ChIP-seq data. And then in part 2, it's more like looking over public available AR data set to get a feeling of some quality control. But as we mentioned, for ChIP-seq, we still want some downstream analysis followed by our part 1. And in lecture, we talk about motif finding. And so this picture is copied from Shirley's slide. So you can imagine TF2 as our AR transcriptional factor. And when we put on AR, if there are some other transcriptional factors that are binding with it or like co-interacting with it, binding to the same DNA region, then since they're so close, even though our antibody only binds to TF2, TF1 and its DNA will be put down as well. So that's how we define co-interacting TFs. And then because if you take a look at our output bed file from part 1, it's essentially just some chromosome start of a PIC region and end of a PIC region. And then we also talk about different motif finding algorithm. Then some softwares will just take a look at those DNA sequence in our given bed file. For example, we might have 1,000 PIC region in our given bed file. And then the software will just take a look at the corresponding DNA sequence and see which motifs are enriched in those DNA regions. So I think in the lecture, we talk about Gibbs sampling, mean, consensus, a lot of different algorithms. And so in question 9, we have 4 different software which are built based on their own algorithm of finding those enriched motifs. And then after finding the enriched motifs, we can find the corresponding transcriptional factors. Is this part clear for you guys? Like why we're doing motive finding? Okay, I assume it's clear if no one is asking any question here. Okay, so there are 4 different software given. We would recommend firstly using Weider because it has user-friendly web page and also it's very fast. So I'll give a live demo here. Okay. So can you guys see the pScanChip screen?
Yes.
Okay, thank you. So for our homework, you can upload your submit SPAD file from part 1 here, like choose file and then just upload. Here I'll just use their sample data as a demonstration, which is K592 cells. Okay, so as I said, that file is just chromosome and start and position of your peak region. And then I think it's fine using the default setting. Maybe for homework, you should maybe change it to HD38 and then just click run. Should be less than 1 minute, really fast. Okay, cool. So you can see now it gives us a list of transcriptional factor names. And if you click on one of them, go inside, you will see like more details about this transcriptional factor. And this is like the PWM matrix talking lecture, but in terms of frequency, it's just raw counts of ACGT here. And here is the SIG logo of the motif. So maybe for transcriptional factor NFYB, it only binds to CCAA TCAG region, DNA region on our genome. So that's what we mean by motif finding and by transcriptional factor finding. And I think, for example, assuming we are inputting AR ChIP-seq here, it just means that for AR ChIP-seq, we can also find these transcriptional factors enriched. So back to our TF1, TF2 figure, if our TF2 is AR, then this would be, a list of possible TF1. So that are like co-interacting together with AR in prostate sample. So this is about this question. Oh, by the way, so this list is already ranked by the global p-value, which is this column. If you hover your mouse over each column, it will show you like the full name as well. So it's already ranked by global p-value, and the higher it is, the more likely it is to be enriched. And you can also download the TXT file if you're interested in any other type of analysis. So again, including a screenshot of maybe the top 10, 15, just a screenshot of this page is fine. You don't need to click in and check, every TF. I was just doing that for showing you it's actually motif. Okay, so this is for this question. Actually, I can also show you Systrom because it's a tool developed by Shirley's lab as well. I think, okay. But I would still recommend using Weider because if you want to use Systrom, you will need to subset for the top 5K lines of your bed file, which is an extra step. But if you're interested in, you can always do it. So if you ever use Galaxy, you might find this interface very familiar because Systrom is built based on Galaxy. And we first want to import data on the left. So you click on import data, upload file. File format would be bed. And then you can just choose the top 5K of your part 1 question. And then just using all default setting and hit execute, then the file will get uploaded. Because it will take a while, I did it in advance. So you can see top 5K.bed is already uploaded. And then we are interested in motif enrichment. So you can search for motif on the tool searching, on the searching space. And then we're actually using SeqPOS motif tool. So find motifs from given regions enriched near the centers. So you can see here, like at most 5K lines, that's why I say Weider might be a better option because you don't need to do any subset. But if you want to use Systrom, you can subset by, either by fold change, the most, the one has highest fold change, or maybe by FDR. Those information are available in .narrow peak file. So, but as I said, that will be an extra step. And you can just click Systrom because we're not interested in de novo motif finding here. And again, human, and hit execute. So this step would take maybe more than 10 minutes. And I can just directly show you the result. You can click on the HTML output. So again, if you click on any one of them, by the way, all of these are transcriptional factors that are fine to be enriched from our BAT file. Again, you will see this Seq logo, and if you click on Show PSSM, you will see like the PWM-like matrix. It might be like transformed or normalized further compared to the original PWM. But again, you can see all those type of data. And since we're doing AR ChIP-seq, you should at least see AR at somewhere, right? So AR is here. And I think same thing for if you're using Weider, you should be able to see AR somewhere. Okay, so that's for question 9. I hope everything is clear. And then maybe we can go question 10. Oh yeah, so look at AR binding. Does AR, this transcriptional factor, mostly binds in promoter, exon, intron, or intergenic region? Also look at QC motifs. So this will bring us back to Systrom DB. Again, you can just choose a data set with really good quality. I'll just pick the second one. And then once you click on it, go to the very bottom. We will first see QC reports, and here's a line, % peaks in promoter, exon, intron, intergenic. Then you will know where AR mostly binds to. You can look at maybe multiple data sets and then see where AR most frequently binds to, and then just give us an answer. It's also like free response, so just put whatever your thoughts here. And then for the QC motifs, it's right next to the QC report. So again, you can see it's very similar to the Systrom output, but this is from publicly available data sets and from previous literature. So for AR ChIP-seq, you can see they are like NR3C1, Fox family, so a lot of this type of transcriptional factors. It's kind of like a double check and validation of your question 9 result, because you should get a very similar result as some public data sets result, right? Okay, so now let's go back. Yeah, so you asked, do you see similar motifs here as those you found in your motif analysis? You should, otherwise it's problematic. And again, just maybe take a screenshot of the QC motif, public data sets QC motif you're looking at. Okay, so I think that's for part 3. Do we have any question here? So now let's talk about part 4 of the homework. So in question 11, we're actually looking at a publicly available RNA-seq data sets, not only for prostate tumor, but also for a wide range of samples. And we want to know like whether the TF family member and AR have actual correlation and expression relationship. So I guess I'll give a live demo of timer. Yeah, and we would recommend using timer for this question. So first, we want to take a look at the gene expression of AR, because all previous analysis was based on ChIP-seq. But in this case, we want to take a look at integrating the analysis with RNA-seq data. So first, let's go to gene D, which will show us the transcriptional factors actual gene expression level in different samples. So as you can imagine, if a TF is really very important for a disease status, it should have either very high or very low expression in different disease versus normal case, right? Okay, so now let's take a look at AR. And since our sample is prostate sample, it's PRAD. Okay, so let's go here. So you can see AR is actually has lower expression, and 1 star means p-value. Oh, sorry, 1 star means p-value is smaller than 0.05. So we can see that for AR, it has lower expression in the PRAD in the prostate sample, and that's for checking AR gene expression only. We also get a list of enriched motifs and enriched TFs that might be co-regulating together with AR. And we can check more of those in gene correlation. And what do we mean by family, right? By TF family. So let's take a look back to the public QC motifs. So here I'm using the publicly available data set, but maybe for your homework, you should use your result from question 9 or something. Okay, so for example, we can see FOXA1, FOXA2, and FOXC1, they are all in Fork's head domain family, and they are found as enriched TF in that public data set. You can also look at multiple data set to get different TF families. Okay, so now let's put our interested gene is, our interesting expression of TF is AR, and we can put FOXA1, FOXA2, and FOXC1. Okay, by the way, remember to click on the purity adjustment because the reason is explained in the homework note. Okay, so now let's go and hit submit. And as I said, this is public data, RNA-seq data sets for all kinds of tissues. Our tissue for homework 3 is prostate, which is PRAD. So let's go down and go all the way to PRAD here. So as you can see, even though all 3 of those TFs are identified as enriched TF in the public data set, only FOXA1 has like very high correlation in terms of gene expression with AR. And let's click on the number. It will actually give us a plot about the correlation map. So the y-axis is the AR expression level in terms of log2 TPM, while x-axis is the TF expression level. In this case, it's FOXA1, and each dot is the data available in this public RNA-seq data sets. So as you can see, for FOXA1, it's almost linear correlationship in terms of gene expression. And let's take a look at FOXA2. Okay, so as you can see, for FOXA2, it's like almost no relationship at all. And even though they are all FOX family and they're all identified as enriched motif, but maybe only FOXA1 is the actual co-regulating factor with AR. And why is this important? Because as Shirley has mentioned in the lecture, there might be some downstream experiments that you want to do based on the ChIP-seq result. For example, we might be interested in which FOXTF that we can knock down in cell line or maybe in animal model, and see how it would affect the expression of other genes and downstream products. So if we have very limited resources, we would definitely want to select the TF that's like actually co-regulating and then knock them down or upregulate them, instead of, you know, randomly testing a wide range list that we get from the motif enrichment, right? So the RNA-seq part is really like a way to further check on the correlating and co-regulating TFs. Okay, so for the homework, since I already did FOXA1 example, maybe you can choose some other TF family that you can observe from your result in Q9, Q10, and then just provide us a screenshot of maybe this page and maybe of the correlation map as well. And it's like a free response question, and it's more for you yourself to explore and, you know, learn from the process. Okay, so now let's go back. Okay, cool. So that's for question 11. And question 12 is our last question. So, you know, for all previous questions, our TFs, our transcriptional factors were obtained from motif enrichment and then find corresponding TF. But there is a way that you don't even need to look at motif enrichment. You can just directly check other publicly available ChIP-seq result. For example, our is AR ChIP-seq. The public dataset probably contain all possible TF ChIP-seq result that you can imagine. And if another TF ChIP-seq signal has very high overlapping region with our AR ChIP-seq BAT file, because again, in BAT file, it's just a list of regions of where your peaks are, then we can say maybe the two TFs are very highly correlated, and maybe they're actually co-regulating. So you can also think of Q12 as a way of validating Q11 results. Let me do a live demo of Systrom Toolkit first. Okay, so let's go to the very bottom, and the default setting is fine. Maybe you want to go with top 10K picks according to peak enrichment, and then just submit the file you get from question 4, the differential picks you get after comparing tumor versus normal prostate sample, and then click submit. So I don't want to directly give out answers here, so I'll use a fake figure that's not homework 3 data. So this figure is copied from Shirley's lecture slide, and assuming it's AR ChIP-seq BAT file, and then, you know, we would see NaNOG has a lot of expression, has a lot of regions overlapping with AR. That means NaNOG is probably, you know, one of the TF that's co-regulating. But again, this is a wrong figure. I'm just using it as an example here. So back to what I said, it's a good way to check your Q11 result because, for example, we have seen that FOXA1 has very high correlation with AR in terms of RNA-seq gene expression. So here you will probably see FOXA1 appearing, but not seeing FOXA2 appearing as some, you know, top TF that has very high correlation. And there will be multiple outputs. This one is static, is dynamic figure because when you hover your mouse over the point, you will see more details about each single dot. You can just take a screenshot of your final result and then maybe include some of your thoughts of how do you think they validate your findings in Q11? Do you think it makes sense? And so on. So again, it's like a free response question. So that's all about part 1 to part 4 of homework 3. We will cover the rest part 5 and part 6 in next week's lab. But if you guys feel you can finish it earlier, because I think after this lab, it shouldn't be that hard to do part 1 to 4. Let me know, and maybe I can try to pre-record next lab and release it earlier, maybe around Friday or something. So you can do the next part of the homework with guidance earlier as well. And lastly, I want to talk about the 2 bonus questions. So the first bonus question is about finding a data set with batch effect and then rerun homework part 1 analysis. Because I'm not sure if you guys noticed, actually for your batch effect data, it's artificially synthesized. I think Dr. Liu and some TAs have tried different publicly available data sets, but none of them have very good batch effect result. So if you know some data with good batch effect, you can maybe include it and then redo the analysis of homework 2, part 1. Okay, so that's for bonus 1. For bonus 2, it's about rewriting all the machine learning questions, PCA questions in part 2 to sklearn in Python. We are thinking about this because Python is a very widely used language now, also for bioinformatics. And sklearn is like the most standard package for doing machine learning. It has a lot of great built-in functions and mathematical equations explained well on their documentation. So we just feel it might be something nice to learn about for future students. But unfortunately, we as TAs, we cannot give you much guidance on the bonus questions because it's more like you are redesigning the homework for future students with your contribution, and it's very freestyle, free response based on your knowledge. And I think doing the bonus question will actually take a lot of time. So I would definitely recommend you to maybe try to finish your homework first. And if you have some extra time, then you can maybe pick one of them and then work on it. And again, if you meet any question during the process, let us know. I mean, not for bonus, but for all kinds of questions in this homework. And again, do not work on homework 3, part 5 yet, because we have not finalized the data set. We will make an announcement ASAP once we have all data sets finalized. Okay. And in the end, it's Q&A time. So I would summarize some questions during the lab. The first question is that, is the bash script is the bash script required for homework? I would say it's not required, but it's relatively easy to use because you just need to make a .sh file and then paste this header at the beginning of your file, and then just paste the rest of the normal code. Also, I think both Harvard and many other institutes are building their server on Slurm. So it's a very nice thing to learn about and try it during this semester, such that maybe it's much easier for you to use them in the future, right? But again, it's not required for the homework. You can just, as the student question, you can just type in terminal and then do the analysis. That's fine as well. And there's also a question about the BWA. Is the index built? Yes, the index should be already available under homework 3 folder. Please do not build your own index because it's very large and it will take you a lot of time to build it. There's also a question about, I think, questions 3 and 4. Like, what's the prefix of your output? It's just, if you take a look at the Copic documentation, you output capital name and underscore blah 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 dot bad dot narrow pic. So that capital name can be replaced with whatever you put here. For example, if you put Q3 or Q4, then your output will all start with Q4, Q3, and underscore with their predefined file name. Okay. And another question is about question 9. So it's a graduate only version, but I think in question 10, it also asks, do you see similar motives here as those you found in your motive analysis? To be honest, even if Q9 is a graduate student question, you probably need to do Q9 in order to make a comparison here. But we will discuss about Q9, like its graduate student version in our TA meeting, and we will let you know. But I think you will probably need to do Q9 in order to answer Q10 for undergrads. Okay, so that summarizes all for this lab. If you have any other question, don't hesitate. Always feel free to let us know, post in the public channel like homework 3 or homework questions. And okay, I hope you all the best with homework 3, and bye-bye.